Hello, 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 TAFE teachers. Hello, fellow students. And hello, subscribers and anyone else who may want to watch this. And my name is Rachel and nice to meet you. Okay, so, and the noise in the background is my rabbits digging at their boxes. I'm really sorry about that. Okay, so this video is video 2 of 2 of my plant identification horticultural TAFE assessment task. Okay, and this is about using a dichotomous key here to identify my plant to its specific scientific name. Okay, this is not my usual content here on my video, and I'm not offended if any of my subscribers do not want to watch this. This is a lengthy video in regards to keying out the Australian native Banksia spinulosa using the dichotomous key PlantNet New South Wales Flora online system. And this is my beautiful Banksia spinulosa here that I am going to be keying out. And this is video 2 of 2 for my practical oral assessment for the TAFE New South Wales Plant Identification Horticultural Course Unit. Okay, so if anyone has any feedback, comments or advice, please comment below. Thank you. Firstly, the dichotomous plant key is used when we have an unidentified plant specimen. We start at, well, it can be used. We start at number 1 on the key. It gives us 2 choices. Then whichever one we pick, the key tells us which number to go to next. Now, I will go through all of this in just a second. I just got a few notes I need to read first. Eventually, if done correctly, we should arrive at the destination of what the exact scientific name of the species is for our plant specimen. For me, this was not easy. I made the mistake of thinking I could grab any plant, take it to the key for identification. Let me tell you, I did not land at the correct scientific name for the flowering of a cherry blossom plant specimen. Not at all. I felt silly when realising that PlantNet New South Wales Flora Online is for Australian plants only, even weeds and grasses. That's why it is called New South Wales Flora Online, because it is about identifying flora in New South Wales. So delving more into facing this assessment, I needed to find 3 plant specimens, photographing the specimens in flower, their leaves and fruit. This was challenging as many plants do not contain all 3 aspects at the one time. I waited months for what I thought was an Acacia longissima to fruit. Checked it daily after it flowered, but all I spotted was wasp galls. Disappointed, I decided this would have to be an Acacia longissima, as further research led me to learn the species rarely seeds. I felt this would not satisfy TAFE requirements, so I finally found another specimen to include in my assessment. It was flowering, fruiting, all leaves present. This actually was Hackia dactyloides, and this isn't the one that I'm identifying today, but I did eventually find a 3rd plant. So this is great, but this is in my written assessment. Yeah, and I will also, I also identified Banksia ericifolia, which is in the assessment as well. But for this oral presentation, this is going to be about the Banksia spinulosa. I had many discussions with TAFE teachers about this keying process because at times I got very stuck. I found out that there is a backwards method that many students use in order to find the correct key route. Then from going backwards, we learn how to go forwards. To do this, it meant finding the plant species name up front, either Plants app, Google Lens, or taking, Plant Snap, Google Lens, or taking the specimen to a nursery. Then looking up online what genus and family the plant specimen fits into. There is only one key for the species and genus, however, for that actual plant specimen, with its scientific name and for its genus. However, when you get to the family, there will be quite a few different routes you can actually pick. And this is where you need to get your learning on about the botany and find the most suitable one, which is, for your plant family. This is not cheating, as to understand a deep level of botanical explanations, terms and descriptions, one would need a degree to understand the terminology. Therefore, to pass this assessment, which is an introduction to plant keys, us students must do what we can, being first year student, to accomplish this. And let me tell you, I definitely learnt along the way using this method. I did try the other method, but it just was not working for me, and it was just taking months. So it's now time to key the Banksia spinulosa specimen I have here. The first specimen, the first few choices I may not fully read out, as the plant is strongly and evidently the other choice. So let's get started here. All right, so what we have to do, so once you get to this page, okay, we go up here. If you can see up here, we go to identification keys and we click on that. Okay, and now we go all the way down and we press on this one, class 7 Magnoliopsida, which is all flowering plants, and we click on that. Okay, now this took, that even took me a while to get my head around this. Okay, so here we go. We've got our choices starting at number 1. Okay, so the two choices here are carnivorous plants versus plants not carnivorous. So I'm actually going to pick plants not carnivorous, because there's no way that my Banksia spinulosa eats other plants. So we click on this and we go down to 4. And then after we go to 4, water plants or plants neither floating on nor growing in water. Definitely the second one, because my plants are not floating. Oh, my plants are not water plants. So yeah, they're not floating on or growing on water. So we go to 104 here. Okay, we go to 104. Okay, so we've got plants either climbing or epiphytic on other plants, or parasitic on aerial parts of other plants. Nope, that doesn't sound like my plant. It was very much standing alone bush. Plants neither climbing nor epiphytic nor parasitic on aerial parts of other plants. Yes, we choose that one. Second option, 206. We go to 206. All right, so let's go to 206. Okay, so leaves apparently, oh, whoops. Go back, I accidentally pressed. Okay, go back. 206. Leaves apparently absent, often reduced to scales, or leaves present, not reduced to scales. So we are definitely leaves present, not reduced to scales. Scales is what you see on conifers. Yes, scales are usually small flat leaves, such as those, yeah, tiny little leaves on conifer branches. Okay, so we go to 230. Okay, so we've got leaves consistently whorled, or leaves alternate or opposite, or leaf solitary. Our leaves are definitely alternate and scattered along. They're scattered along here in an alternate way. See, they are definitely not opposite. And at the tips, they're a little bit whorled, but definitely up here they're opposite. They're alternate and they are scattered. So we are going to choose 254. Let's go to 254. Okay. Leaves usually with reticulate venation, vascular bundles open, that is secondary thickening occurs, taproot present at least in seedlings, flower parts usually in whorls of 4 or 5, embryo mostly with 2 cotyledons, which are known as dicots. That means when they open the first shoot there are 2 little leaves. Monocots are plants that just have 1 little shoot when they first open. Okay, or leaves usually with parallel venation, vascular bundles closed, that is no secondary thickening. That's usually a second thickening on the stem there. Root systems fibrous. Flower plants usually in whorls of 3. Embryo mostly with 1 cotyledon. Doesn't sound to me like, you know, this is my plant here. I would definitely go the top option here. Firstly, these small leaves have reticulate venation. So even though they're so tiny, okay, I have a magnifying glass. Let me show you reticulate venation on this note here. So if you can see this, so you've got here the prominent veins on the lamina, okay, and midrib of this leaf, and you've got all these little like scattered veins. Okay, this is called unevenly scattered veins on the leaf, reticulate venation. And parallel venation is when they're very straight on the leaf. So this is reticulate venation. Dicots, have reticulate venation. They are plants that when young, the seedlings sprout. They have 2 cotyledons, which is the leaf that I was telling you, the 2 leaves that I was telling you about, the 2 first shoots, I should say. Monocots have 1 cotyledon. It's also, the little shoot. Dicots, which is what my plant is, are also woody. These are definitely woody. And the vascular bundles are mainly always open in woody plants, and the stems have a cambium layer which separates the xylem and the phloem. And the xylem in here actually transports water and the phloem transports food, and when there's a cambium layer in between them, it means that they're open and they get to have a second thickening on their stem, which makes them stronger. And the small tubular flowers have 4 tepals, 4 anthers hidden inside, and an elongated carpel, which is the female part. The style extends outwards, forming the edge of the spike. The taproot is present after 2 years of initial growth, so therefore it is still present on the plant. So definitely I'm going to pick this top one. Okay, so that says go to 255. Okay, either leaves compound with 2 or more distinct pinnae, or leaves 1 foliate or simple, sometimes lamina, deeply lobed but not divided into discrete leaflets. I'm actually going to pick the bottom one here because our leaf is actually simple. They only have 1 pulvinus, which is a small bulbous area where this at the stem and stalk junction, the leaf is not divided into 2 or more leaflets like a compound leaf. So definitely I'm going to be picking 314. Oh, the second option, which leads us to 314. We go to 314. Okay, flowers enclosed in an invaginated floral axis or fruit multiple, a zirconium or fig. We're definitely not that. Flowers not enclosed in an invaginated floral axis, not a zirconium or a fig. Here's a little picture here. And this actual little picture I drew here is actually what the flower of a fig looks like in an invaginated floral axis. It's actually all on the inside. So you don't see any of the flower on the outside. It's all on the inside. Everything blooming on the inside. So it's like a little, it's a little capsule flower. It's zirconium. Okay, so we're going to go to 315. So we go to 315. Okay, here we go. Flowers unisexual, arranged in a cyathium, white latex present, or flowers either bisexual, or if unisexual, they're not arranged in a cyathium, white latex either absent, or if rarely present, they're flowers bisexual. Okay, so when I snipped these branches from my main tree, I noticed there was no white latex, so I doubt the plant is bisexual. Also, it is not arranged in a cyathium, which is like this, where we have the female flower surrounded by all these male flowers. So that's not the structure of our plant there. Okay, our plant is unisexual as it has both female and male parts being the stamen, consisting of the filament and the anther, and the female part being the pistil, consisting of the stigma style and the ovary. So we are actually going to go for a bisexual plant here, which go to 316. 316. Okay, ochreae, which is sheaths of fewed stipules present around the petiole stem. Ochreae absent. So we're going to go ochreae absent. Ochreae is a sheath wrapped around the plant, petiole. There's no ochreae on my plant. So here's the stem. Okay, and here's the ochreae, this little sheath here, which goes onto this petiole here. So it's formed just after the insertion of leaf on stem, formed from stipules. Stipules are small leaf-like structures often present at the base, and these fuse together creating an ocrea. Well, we don't actually have that on my plant, so we go to 317. Okay, 317. Ovary superior or the flower naked. Flower naked actually means there's no leaves on the flower, on the plant. A good example of this is naked ladies. They're pink flowers with a maroon stem and there's no leaves. Ovary inferior or half inferior. So even though we don't have a naked flower, our ovary, I believe, is superior, as it's above the other parts of the lower perianth section, including the tepals. This is also known as being hypogynous. So we're actually going to pick 318. Okay, so 318. Carpels are fused. Oh wait, sorry, just let me read this. Carpels free. Fruit usually separating into distinct carpels, that is 2 or more ovaries present, sometimes carpels enclosed in a hypanthium and ovary apparently solitary, or carpels fused or the carpel solitary, that is ovary and fruit solitary. So the reason I'm actually going to pick the bottom one, because we have yes, there is one ovary in a single structure with the style and the stigma, so it's not 2 or more ovaries. Carpels are fused. So we're going to do the second one because the ovary is in a single structure with the style and the stigma. Okay, so 335. Okay, 335. All right. Fertile stamens, more than twice as many as sepals or tepals. Tepals as in Proteaceae or Moraceae, or fertile stamens to twice as many as sepals or tepals. Now this one really threw me and had me stuck on this for a while, because with all the other routes that I could have picked for the family of Banksia spinulosa, which I will show you that family name in a minute, all the others were mentioning that the leaves were pinnate. And so just let me read this. I did take a note here. So it did have me doing a lot of questioning, as both choices I felt did not suit Banksia spinulosa. However, reading through all the other available key routes, this was best suited as the Banksia spinulosa leaves are not pinnate and the flowers are not actinomorphic. Okay, and that means that the flowers aren't symmetrical. That means the flowers, aren't symmetrical. Pinnate were greatly emphasized in other routes. So I concluded that since the tepals only split into 4 individual parts when the flower fully opens, it would be best to go with the second option, being fertile stamens to twice as many as sepals and tepals, because in the early stage there are 4 stamens to 1 tepal before it splits. So this was the only route that suited, and so I had to find a way to make one of these fit Banksia spinulosa. I mean, if there's another one and someone can shed some light on this, let me know. I know that this key is a work in process, but I actually had to pick this one, knowing that the tepals split right at the end. So in the start, the stamens are more than the tepals. So I picked the bottom one, which leaves us to 356. Okay, so here we go. Oh, and also, even though the top choice has in brackets for the last one, Proteaceae, if I chose the first choice, I wouldn't actually be led to the correct family of Banksia spinulosa. So I'm not sure why that would have Proteaceae in brackets, because it doesn't lead you to Proteaceae. Okay, 356. Okay, so we have either perianth in 1 whorl or the flower naked, or perianth in 2 whorls. So my plant had the perianth in 1 whorl as the tepals enveloped the male and female parts of my plant. So it is the top one, even though my flower is not naked. Okay, so we go to 357. Okay, again, flowers naked, perianth absent, or flowers with perianth present. So it's the bottom one because our perianth is present. This consists of the tepals. They're called tepals because the sepals and the petals couldn't be easily classified. Usually petals are at the top and sepals are at the bottom. They're the green leaf-like structure. So yeah, they get called tepals if they're all like one. And yeah, the sepal is also known as the calyx, which is the lower part, and the corolla being the top part. And yeah, so we have a perianth, so we are picking 360. Okay, we go to 360. So we're getting very close now. I can feel it. Okay, perianth segment scarious, perianth segment herbaceous or petaloid. So we are the bottom one, perianth segment herbaceous or petaloid, because when the we are petaloid because when the perianth is non-green, it's herb, which is yeah, so ours wasn't green, it's called petaloid. So yeah, our Banksia is petaloid as it appeared yellowish around the pistil and stamens. And now just want to mention these flowers, these flowers are not one flower. This is hundreds of small tiny flowers on these bracts on this axis here. So it's very hard to see and dissect and magnify and look into them. So I needed to do a lot of online research about this as well to get real like close up, almost microscopic pictures on these. But I managed to do a lot with a magnifying glass and a torch as well. Okay, so we're the bottom one, so we're going to go to 361. Okay, 361. Okay, so here we go. Stamens 8 to 10 or stamens less than 8. So, okay, this means less than, this means more than, okay? So I go, oh, this might be back to front maybe if I'm recording this. Left ear is less than, right ear is more than. That's the way I remember it. Okay, so we have 4 stamens. So we are going to go with less than, so that's the bottom one. So we go 371. Okay, so 371. Styles 2 or more, or the style branched compared to style simple, unbranched, stigma, sometimes lobed. So our Banksia has a simple style, unbranched, not lobed. It is hooked, however, getting its name as the hairpin Banksia. Okay, so we are going to go to 376. Okay, perianth tubular, at least in bud, or tubular hypanthium present. Perianth or perianth not tubular, sometimes fused at the base and forming a shallow cup. So our one is definitely the top one. We have a tubular, tiny little flower. They're all little tubes along here. And yeah, so the perianth of my specimen is sorry, just a second. Okay, so welcome back. Sorry, I just had someone come to my door. Okay, so I hope that you can all see this. So we are up to, so we're going to 377. All right, so we have stamens always 6, petals 6, calyx represented by a cup which enlarges in fruit, or we have stamens mostly 2 to 5, sometimes 6 to 8, calyx not a cup which enlarges in a fruit. So we have a calyx that is not a cup which enlarges into fruit, as the flowers completely dry up and then the woody bract grows the hard woody follicles known as seeds, so with the seeds enclosed inside them. So we are going to pick 378. well, this one, go to 378. Okay, so it was bottom one, so 378. Upper part of perianth tube falling, the lower part persisting and enclosing the fruit, or otherwise. So literally I'm going to pick otherwise because that top one doesn't seem to describe what happened to my plant. The stamens are, yes, the stamens, the perianth stays very much intact, and stamens are more than, oh, sorry, this is the next one. I'm reading notes for the next one. Okay, so we're going to otherwise, so 379 because the stamens stay intact. Okay, stamens 2 inserted on the rim of a tubular hypanthium, or stamens more than 3 not inserted on the rim of a tubular hypanthium. So we're going to pick 380 because our stamens are more than 3, they're 4, and they're not inserted on a tubular hypanthium, as it just has a tubular perianth. There we go. Okay, 380. Trees or shrubs, stamens opposite to and fused with the perianth segments, or herbs and subshrubs, stamens alternating with the perianth segments. We're going to definitely pick the top one, and that brings us to the name Proteaceae, and that is the family of our Banksia spinulosa. So we're going to click on this. Our specimen is a shrub. I only measured it up to 1.5 meters tall. Some Banksias can actually grow up to 30 meters tall, and these are actual tree sizes. So with our Proteaceae, with our Banksia spinulosa, the stamens were positioned opposite to and all attached with the perianth segments. Okay, so here we go. Also, our plant is an angiosperm, as it's a flowering plant and the seeds enclosed in their fruit. Okay, so here we are, family, Proteaceae. Now we've got to do the key. This brings us to the key of the genera, the genus. So this is the next key, but this won't be as big as the family key. That's why in the family key you've got many options to arrive at Proteaceae. However, this one you will only have one route to arrive at the proper genus. So here we go, let's start at number 1. Inflorescences, 7 flowered heads consisting of a central terminal flower surrounded by a ring of flowers, which in turn are surrounded by conspicuous involucra. Flowers red, fruit a woody follicle bearing 2 conspicuous lateral subterminal horns. Doesn't sound like that. Or inflorescences not 7 flowered, not terminated by a flower, involucra absent or present, flowers variously coloured, fruit dehiscent or indehiscent. Now, by the way, there's a spelling mistake there, TAFE teachers. You need to fix, well, not you guys, but, you know, whoever puts this key together. Anyway, look, dehiscent. It's, see, look, Dehiscent. That's correct, but the indehiscent, they've got the S and the I around the wrong way. That threw me because I actually looked that up to see, and then I couldn't get any spelling, like it wouldn't show up in Google, so that's how I know it's a mistake. Well, I hope it's a mistake. Anyway, okay, so only occasionally horned. So I definitely am going to pick the second one. I have a few notes on this. So, our inflorescences are not, which also means flower, are not consistently 7 flowered, as the spike consists of hundreds. So this is the spike, and it consists of hundreds of flowers on the bract. The inflorescence does not terminate by a flower, as it has flowers opening up in succession. They're continuously flowering right up to the axis. Okay, so then we have, yes, there are hairy little involucra bracts present at the base of the flowers, and yeah, so we are definitely going to pick, and our, yeah, so we're going to pick number 2. Okay, so we go to number 2, Flowers sessile in dense cone-like globose to cylindrical inflorescences or cone fluorescences, the bracts becoming woody in fruit. Well, that just 100% sounds like our plant, doesn't it? Or flowers pediculate to sessile, but not in cone-like globose or to cylindrical inflorescences or cone fluorescences, bracts not becoming woody. Okay, so here we are going to pick the top one because we are in a cone-like globose to cylindrical inflorescence. And yeah, so the flowers they range from a yellow in early stages and they become an orangey a little bit later on, but there are different colors. They can even be black and yellow where the styles are black, and this is why sometimes they're called the hairpin banksia. The fruit is dehiscent, so meaning the pericarp, which is the casing around the seeds, it actually splits. I think these were notes for the other one we did before actually, and they open to release the seeds. But yes, 100% this sounds like our plant, this top one, woody in fruit, bracts, all of this. So we are going to go to 3. So let's just click on 3, even though it's just straight underneath. Okay, so flowering and fruiting cone fluorescence axis, usually up to more than 5 or less than, hang on a second. Oh, just give me one. Oh, I lost my phone. Sorry, TAFE teachers, just give me 1 second. Sorry about that, I just knocked everything over. But we're back. Okay, so let's continue. So top one here in the genera on number 3. Okay, flowering and fruiting cone fluorescence axis, usually 5 centimeters long and with flowers borne in pairs. Fruit persistent, transversely orientated follicle. Leaves usually toothed or less frequently entire. This definitely sounds like my plant. I am just going to pick this straight away. And our genera is Banksia. Yep, so that is the genus for my plant. And yeah, this just sounds perfect. So let's click on this, and then we're going to go to the key of species. So there we go, genus Banker. Look at this Banksia, not Banker. Here we go, look at that. All right, so I'm on the right path. Okay, so here we go to number 1. So with this keying system, there is family keys, genus keys, and then there is species keys. So now we are up to almost at the end. Okay, here we go. Styles straight or gently curved after anthesis, or styles after anthesis hooked just below apex. We are actually going to pick this second one here because the style is the longest growing part of the flower and mostly remains hooked at the apex, which is the tip. So we're going to go to 12. Okay, so losing my voice here. Leaves more than 3 centimeters long, scattered, or leaves usually less than 2 centimeters long, crowded. We are definitely 13 because we have scattered little leaves here up the stem. Okay, and then we are going to go to, okay, so what did I just do there? Oh, so yeah, we go to 13, and the leaves are definitely more than 3 centimeters long. Oh, we'll just come down here for 13. Okay, so leaves 1 to 2 millimeter wide. Yes, we are that. Margins revolute. Yes, our margins here are actually revolute. Now, you have to look really closely with a magnifying glass. It just means that the leaf edge rolls to the underside a bit, so you don't always, it sort of conceals the underside, so you don't always see underneath very clearly. And yeah, so margins revolute, so as the lower surface is mainly concealed. Da, 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 da! Banksia spinulosa. Oh yeah, look, there it is. Hello. Oh, it's awesome. I wonder if we can enlarge this. Let me get my tech on. Zoom. Yeah, look at this. Oh, look at this. Oh, now I've lost it. How to get it back. Anyway, Yeah. Oh, I zoomed it a bit. Look at this. There we are. Now, the one that I originally had, which was on this plant here, was this beautiful orange colour. And then as time passed by, actually turned to a deep orangey red. But to start with, this hairpin banksia that I first, this plant that I just kept going back and forth to had these beautiful yellow Banksia spinulosa flowers. And then this one is also a Banksia spinulosa, but this one has the black styles, and this is one that is known as hairpin Banksia. Well, yeah, it's the same plant. It's just they have different slight variances in colour. Anyway, that's it. Automatic pass. What do you reckon? Because this is not also an... Oh, sorry. Let's be professional. Communication skills. This isn't just an oral presentation. This is a physical, verbal. That's oral. Yeah. This is also physical and picturesque. Okay, so... Okay, no. But yes, please pass me on this assessment so I can move on. Nice to meet you all, even though you met me, and I